Today we're going to talk about meiosis and how meiosis allows us to have sex, which is important because we're sexual, so to reproduce, we've got to be able to produce sex cells. So let's talk about how you start your life, right? You started your life when a sperm fertilized an egg, and that process is called fertilization, you became what's called a zygote. And then you underwent a series of mitotic divisions, right? Where that cell divided and divided and divided and got bigger and then developed. It turned on different genes so that you became heart tissue, nerve tissue, skin tissue, all the organs in your body. And now you're a mature adult, right? So at some point you've got some organs in your body that need to produce sex cells, right? They're called testes and ovaries. So there's a process that goes on in those cells called meiosis, okay? So meiosis is a process where these cells undergo a special kind of cell division and they produce sex cells. Now, sex cells are what we call haploid cells. They abbreviate that with the symbol N. Haploid means they have half the chromosome number. Body cells are called diploid cells. The abbreviation for that is 2N. So body cells have 46 chromosomes. Every cell in your body, except for sperm and eggs, have 46 chromosomes. Sex cells have half that number, 23 chromosomes. So this process of meiosis is basically taking diploid cells in your in your testicles and ovaries and producing sex cells. Okay. Now, an easy way to remember meiosis as opposed to mitosis is, remember, mitosis occurs in my toes. So if I stub my toe and skin it, those skin cells are going to grow back through the process of mitosis. So my toe heals itself through mitosis. Meiosis occurs in my huevos. So if you remember your Spanish, huevos means eggs, so that's where eggs are produced in the ovary. Or another term for eggs, or huevos in Spanish, is balls, testicles. So this process also occurs in the testicles to produce sperm cells. Okay, so a quick overview. Meiosis, like mitosis, always begins with replication of chromosomes. So we form two sets of sister chromatids for each chromosome. Then we pair up our chromosomes into what are known as homologous pairs. We have 23 of these pairs of chromosomes, and we separate them into two sets, each with half the chromosome number. Then we still have our initial sister chromatids, so we have to go through a second cell division in meiosis to separate those sister chromatids. And when we're done, we've got sex cells with the haploid number, with half the chromosomes from the original, because we paired all of our 23 pairs of chromosomes up and we divided them. So that's how meiosis occurs. Now, meiosis and mitosis, it's hard to kind of understand how these are really different. So let's look at them and line them up right next to each other. So they both begin the exact same way, right? We replicate our chromosomes into sister chromatids. So prophase 1, they both have chromosomes forming, and those chromosomes exist as sister chromatids, right? Those little X's that you see there, okay? But there's one difference in prophase. Notice in meiosis, they are paired up in what we call homologous pairs. Homo means the same, so these are, say, pair number 1 and pair number 2. Whereas in prophase of mitosis, they all just appear. All those chromosomes just appear anywhere, okay? And in metaphase, it's similar, but it's different. Notice in metaphase of mitosis, all 4 chromosomes are lining up along the spindle, and they're all going to be separated. But in metaphase of meiosis 1, they're lining up as pairs, and we're separating homologous pairs here. Okay. Then in anaphase and telophase, we're just separating and reforming our chromosomes in mitosis. So we have 2 cells that are identical to the parent cell in mitosis. Okay. But we have in meiosis a second set of cell divisions. Why do we have that? Well, because we need to separate these sister chromatids. We haven't yet separated them yet. So the second meiotic division, which is called meiosis 2, is where we're separating the sister chromatids to form 4 haploid cells. And again, the symbol we use for that is N. So mitosis, we're producing 2 diploid cells. Meiosis, we're producing 4 haploid cells. So that's the important difference between mitosis and meiosis. Okay, so now let's break down meiosis into those 2 steps, meiosis 1 and meiosis 2. So in meiosis 1, first in interphase, we see the nucleus, and then in prophase, chromosomes form. But again, notice how they're forming in these homologous pairs. Because each pair has two sister chromatids, we've got two chromatids and two chromatids in each of the pairs. So we call those tetrads. Remember, tetra means four. So a tetrad is just four sister chromatids from each homologous pair. Okay? Then in metaphase, we line up the pairs, and in anaphase, we separate those homologous pairs. So in meiosis 1, basically what we've done is we have separated homologous chromosomes, okay? Those two. Now let's look at meiosis 2. Remember, meiosis 2, we've separated homologous chromosomes, and technically we have chromosomes with half the chromosome number, but we still have chromosomes that have sister chromatids, right? We still haven't separated the sister chromatids. So again, this is, meiosis 2 is about separating sister chromatids. So in prophase, chromosomes are already there, so not much has changed. Metaphase, we're lining up each of the chromosomes along the plate, much like mitosis. Anaphase, we're separating the sister chromatids, again, much like mitosis. And then in telophase and cytokinesis, we're pinching them off into, again, a total of 4 haploid cells. So that's the result. Now, one difference in males versus females is sort of a quality versus quantity. So males start with 1 diploid cell, and they produce 4 haploid sperm cells. They divide up the cytoplasm evenly and produce 4 very small sperm cells. Now, when females produce eggs, they start with 1 diploid cell, and then that divides, and it divides again. So chromosomes are divided up exactly the same. The meiotic process is the same, but notice the process of cytokinesis, of splitting up the cytoplasm, doesn't exist when we form eggs. All the cytoplasm is staying around 1 set of chromosomes. So we're going for a larger cell, a cell that can basically survive that ovulation period of traveling from the ovary down the fallopian tubes and into the uterus. And if it gets fertilized, it can even start to maybe even divide and start to grow before it gets fed by those blood vessels in the uterus. So again, sperm, men produce billions of sperm throughout their life. We're basically going for quantity. Females only produce maybe 500 eggs in their life, so they want to invest more in them. They want them to kind of make sure that if they do get fertilized, they can be viable and they can survive that journey down into the uterus and the process of fertilization. One difference, or one other thing about meiosis that's important is we create different combinations of our chromosomes. So let's say we only had 2 pairs of chromosomes. We have 23, but if we only had 2, we could divide it, let's say the red ones are the ones you got from your mom. Well, they could both go and divide into that one, and then they could divide into these eggs, and we could have that. And then the blue ones you got from dad could go there, and then they could go down here. Or we could get one from mom and one from dad, one from mom, one from dad. There's 2 possibilities here, right? So there's actually, in this case, there's 2 to the second combinations. Now in us, we've got 23 pairs of chromosomes, so we actually have 2 to the 23rd combination. That's a lot. That is about 8.6 million combinations of sperm or eggs. So the chances that you and a sibling are identical in terms of all the chromosomes is almost 0, right? So we just have so many different chromosomes that we're separating during meiosis, and then the possibility of getting the exact 2 from mom and dad, even though they're the same people with the same set of chromosomes, is almost 0. So that is important because that means that we're all genetically unique. We all have genetic diversity. Something else that allows for genetic diversity in populations is this idea of crossing over. Now, crossing over, when the tetrads form during prophase 1, chromosomes are kind of sticky. And it's kind of like doughy, sticky things. When you put them together, they stick together. And then if you pull them apart, well, notice when we pull these apart, we've got a little bit of red there and a little bit of blue there. So we've mixed up some DNA from these homologous chromosomes. So maybe I had dad's chromosome, and now it's got chunks of the chromosome I got from my mom on it. And now that those sex cells are going to have new combinated spliced together chromosomes. So we're really even mixing up our DNA even more in this process of meiosis, because crossing over, which occurs in about 10% of our homologous chromosomes is kind of cutting and splicing together the homologous chromosomes to create new and different combinations. Again, more genetic diversity, and genetic diversity is good. It means that we're all going to be different. We're all going to have some different attributes or adaptations. So this is kind of an overview of meiosis and why meiosis is important in creating sex cells.